In this tutorial, we will demonstrate how to export data from InSilicoDB to GenePattern and create a heat map with that data. InSilicoDB is an open platform for managing and analyzing genomic datasets. It contains thousands of public samples and provides connections to analysis tools such as GenePattern. More information about InSilicoDB can be found at their website, insilico.ulb.ac.be. We'll start by browsing the public datasets. For this tutorial, we'll use the GeoDataset GSE4635. Click Edit Show Clinical Annotations. Next, we need to select a curation or create a new one. For this tutorial, we'll select the existing geo annotation. Once this has been selected, we can close the window. We can now export this dataset to GenePattern. To do so, click on the black arrow next to the GenePattern icon and the word Export. This will display the export options. We will leave the options at their default values and click Export. If you are not already logged into InSilicoDB, you will need to do so now. Once logged in, you will be prompted for your GenePattern public server login. The data will then be sent to an import module in GenePattern, and you can open GenePattern to see your job as it completes in the job result page. Note that you will need to refresh the browser to see progress. Now that the job has completed, all of the clinical annotations from InSilicoDB are now available to use in GenePattern. Let's first save the phenotype of interest. We can then open comparative marker selection on our expression data by clicking the arrow button next to the GCT file and selecting Comparative Marker Selection from the list that appears. Comparative marker selection is now displayed with the GCT file already filled in. We now need to specify our CLS file. To be sure we get the CLS file we want, click Specify URL and drag the smokers.cls file from the Recent Jobs panel on the right. We'll also need to choose Yes from the drop-down for the log transformed data parameter, because all FRMA renormalized gene expression datasets in InSilicoDB are log transformed, and that was the normalization we chose when we exported from InSilicoDB. We can now run comparative marker selection. Once that is finished, we can send the resulting ODF file to extract comparative marker results. Before that can be run, we must specify the GCT file from InSilicoDB as our dataset file. We'll also set the field to filter features on and the minimum value for that filter. Once that is done, we run the module. When the job completes, we send the GCT file to heatmap viewer. The heatmap viewer will now launch. We can then change the size of the heat map by going to View, View Options, and moving the sliders to adjust the size. We'll now label our samples using the phenotype file we saved from InSilicoDB. To do so, select Label Samples from the File menu and browse to smokers.cls. The yellow samples are non-smokers and the red samples are current smokers. You can click the colored bars to read labels and change their colors. Let's make one last adjustment and sort our features. From the View menu, select Sort Features, choosing the smokers.cls file, Signal to Noise, and Sort Ascending. We now have a heat map of overexpressed genes, which came from a public geodataset exported from InSilicoDB and analyzed in GenePattern. For more information about InSilicoDB, please visit insilico.ulb.ac.be. For more information about GenePattern, please visit genepattern.org. You can also find us on Twitter.